Hello again. Today we're going to talk about C3 plants and how they have evolved to overcome the problem of photorespiration. Now we know that in plants, exchange of gases in the form of carbon dioxide or oxygen or water takes place through the stomatal pores. And it is through the stomatal pore where carbon dioxide enters into the mesophyll cells of the C3 plants, and this carbon dioxide is then fixed in the Calvin cycle by the enzyme Rubisco. So this we are aware. But what really happens when you have very, very high temperatures during the day? So when you have very high temperatures, it's very, very hot, what happens is the plant tries to reduce the transpiration rate and the loss of water through transpiration. And one mechanism by which it does that is to close the stomatal pore. Now when it closes the stomatal pore, atmospheric carbon dioxide cannot enter into the mesophyll cells of the plant. But also what we have to understand is that because you still have light, the light-dependent reactions of photosynthesis will continue to take place, and as long as the light-dependent reaction continues to take place, you will also have the evolution of oxygen. So what happens is you then slowly start having a buildup of oxygen inside the mesophyll cells of the plant, and this oxygen has nowhere to go. Because the stomatal pore that you have over here, this stomatal pore is closed. And although the carbon dioxide which is inside the mesophyll cell, this keeps on entering the Calvin cycle, but there is no new entry or no new influx of atmospheric carbon dioxide. So when this situation occurs in the plant then undergoes what we call as the Warburg effect. And in Warburg effect, what happens is when the concentration of oxygen is high and exceeds the concentration which is normally found in the plant, which is around 0.5%, then the photosynthetic efficiency of the C3 plant starts to drop. And this phosphoglycolate is then converted to glycolate in the chloroplast, and then this glycolate cannot be immediately utilized by the plant. So what it does is it shuttles the glycolate out of the chloroplast into another organelle, what we call as the peroxisome. Now in the peroxisome, glycolate is then converted to glyoxylate, and this glyoxylate is then further shuttled outside of the peroxisomes into the mitochondria, and in the mitochondria, glyoxylate is then converted to glycine. And this glycine is then either exported out towards the phloem, or it is then converted by serine hydroxymethyltransferase, converted into serine. So glycine is converted to serine in the mitochondria. From the mitochondria, serine can either be transported outside, or it is re-shuttled back to the peroxisome. And in the peroxisome, it is finally converted to glycerate. And then this glycerate finally enters back into the chloroplast, and in the chloroplast, this glycerate is then converted back to your 3-phosphoglycerate, which is actually the first product or stable product that you get from the Calvin cycle. So what we can see is that instead of the plant getting 3-phosphoglycerate directly from binding of carbon dioxide, it has to undergo a lengthy process wherein compounds are shuttled back and forth from the chloroplast to the peroxisome, from the peroxisome to the mitochondria, from the mitochondria back to the peroxisome, and then finally to chloroplast, to give you the same 3-phosphoglycerate, which then can be utilized in the carbon cycle. Now when this happens, then what you get is a decrease in the efficiency of the photosynthetic output, and that is what is reflected over here, which we term as the Warburg effect. So as a consequence of the process of photorespiration, which takes place in C3 plants at very high temperatures and increasing light intensity, what you have is that the photosynthetic output decreases by around 25%. So what you would normally have as 100% photosynthetic efficiency under normal circumstances, that photosynthetic efficiency starts decreasing to around 75%. So there is a huge drop in photosynthetic efficiency due to the process of photorespiration, which happens because of the increase in the oxygen concentration in the internal tissues. And this happens because primarily the Rubisco enzyme, which catalyzes the binding of carbon dioxide to RUBP, does not have a preference for carbon dioxide, and hence therefore it also catalyzes the binding of oxygen to RUBP, which leads to the production of phosphoglycolate. So what the C3 plants have evolved is that in order to overcome the problem of photorespiration that you find in the C3 plants due to the elevated concentrations of oxygen in the internal tissues, the plants evolve an enzyme called phosphoenol pyruvate carboxylase, and this enzyme preferentially binds carbon dioxide to another substrate called phosphoenol pyruvate. And oxygen cannot bind to phosphoenol pyruvate because the enzyme phosphoenol pyruvate carboxylase preferentially binds carbon dioxide to phosphoenol pyruvate, unlike the enzyme Rubisco, which is RUBP carboxylase oxygenase, which can bind both carbon dioxide as well as your oxygen to RUBP. So here, once your carbon dioxide is bound to PEP or phosphoenol pyruvate, you have the formation of a compound called oxaloacetate, and this oxaloacetate is a 4-carbon compound, which is a stable product of this process of carbon dioxide fixation. So therefore, this pathway of carbon dioxide fixation is called as the C4 pathway because the first stable product, oxaloacetate, is a 4-carbon compound. Now this oxaloacetate then gets converted to malate, and then malate will then release the carbon dioxide into the internal tissues of the plant, where it can then enter into the carbon cycle. So the question now that might occur to some of us is that, well, we have oxygen over here and we have carbon dioxide over here. Oxygen may not bind to phosphoenol pyruvate, but then oxygen may bind to the RUBP, which is present over here before it enters into the carbon cycle. So how do the C4 plants manage to overcome this problem? Well, they do that by having an evolution in the anatomy of the plants. Well, what we see is that in the C3 plants, the entire process of the light-independent reaction of photosynthesis takes place in the mesophyll cell chloroplast, or takes place in the mesophyll cells itself. But when you look at the anatomy of the C4 plants, what we begin to see is that the initial process of carbon dioxide fixation takes place in the mesophyll cells, or the chloroplast present in the mesophyll cells, and the Calvin cycle takes place in chloroplasts which are found in the bundle sheath cells. So the anatomical adaptation of C4 plants compared to C3 plants is that the C4 plants now have chloroplasts in the bundle sheath cells and also have chloroplasts in the mesophyll cells. In the mesophyll cells, you have the initial fixation of carbon dioxide, and in the bundle sheath cells, you have the Calvin cycle. And the carbon dioxide which is fixed in the mesophyll cell chloroplast, that carbon dioxide is shuttled to the bundle sheath cells, and here the carbon dioxide is then assimilated into the Calvin cycle. So when you look at the anatomy, as I said earlier, what you see is that in C3 plants, the bundle sheath cells that you have do not contain any chloroplasts. But in C4 plants, the mesophyll cells have chloroplasts, but also the bundle sheath cells that you have over here, they also have chloroplasts in them. And this particular anatomical feature is what is referred to as the Kranz anatomy, where the bundle sheath cells contains chloroplasts and it looks like a crown embedded with precious stones. This anatomical adaptation of the C4 plants allows the plant to overcome the problem of photorespiration. So how does it do that? Well, it does that because if you look at the electron microscopy images of the chloroplasts, which are found in the mesophyll cells and in the bundle sheath cells, we see that the chloroplasts in the mesophyll cells have the grana in them, whereas those that are present in the bundle sheath chloroplasts, do not have grana, or even if they have grana, the grana are not developed. So what does this tell us? It tells us that in the bundle sheath chloroplast, you do not have the light-dependent reactions of photosynthesis, because the light-dependent reaction takes place only in the thylakoid membranes of the grana. And why does this happen? It happens because the bundle sheath cells are located further inside of the leaf tissue. And because they are located further inside of the leaf tissue, the chances of light penetrating into the bundle sheath is very, very less. And when light cannot penetrate into the bundle sheath cells, then having the grana in the chloroplast makes no biological sense. It is of no biological advantage to the plant. The second point is that if you do not have the light-dependent reaction taking place in the chloroplast of the bundle sheath cells, then you do not have the production of the oxygen. And when you do not have production or evolution of oxygen, then here this tissue is oxygen deficient compared to the chloroplasts that are found in the mesophyll cells, where you have the light-dependent reaction of photosynthesis generating oxygen as a byproduct. So when you compare the two types of chloroplasts, the mesophyll cell chloroplasts will have higher concentrations of oxygen compared to the bundle sheath cells of chloroplasts, which are oxygen deficient. So how does this deficiency in oxygen help to overcome photorespiration? Well, what you have is that in the C4 plants or the C4 pathway, because you have the chloroplast in the bundle sheath cells and chloroplast in the mesophyll cells, what you have is that you have a case of uptake of carbon dioxide, or the initial fixation of carbon dioxide is separated in space from fixation of carbon dioxide in the Calvin cycle. So the atmospheric carbon dioxide enters into the mesophyll cells, and when it enters into the mesophyll cells that you have over here, this carbon dioxide is not fixed by Rubisco, but rather it is fixed by your phosphoenol pyruvate carboxylase. And what phosphoenol pyruvate carboxylase it does is it converts your phosphoenol pyruvate into oxaloacetate, which is then finally converted into malate. And this malate, from the chloroplast that you have in the bundle sheath cells, this malate is then transported out of the chloroplast into the bundle sheath cells. Now, when it is transported into the bundle sheath cells, this malate that you have, which is a C4 compound, this malate is decarboxylated, which means the carbon dioxide is removed from malate, and this carbon dioxide then enters into the Calvin cycle in the chloroplast of the bundle sheath cells. And the pyruvate, which is regenerated, this is shuttled back to the chloroplast, where it is converted to phosphoenol pyruvate, and this phosphoenol pyruvate can then fix atmospheric carbon dioxide to give you malate, and this malate can then be transported back to the bundle sheath cells. It becomes decarboxylated, and this carbon dioxide can then enter into the Calvin cycle. And the advantage of this process is that whatever carbon dioxide that you have over here, this does not have to compete with oxygen, because this oxygen is at very low levels, if present at all, is at very low levels in the bundle sheath cells, because these chloroplasts do not have the grana to undertake the light dependent reaction of photosynthesis. So you have a case where the carbon dioxide now does not have to compete with oxygen, and therefore the problem of decrease in photosynthetic efficiency due to photorespiration does not occur. So this is the adaptation that you have in the C4 plants, and the movement of these molecules is facilitated through the plasmodesmata, which are the connections between these different cells that you have in the tissue of the leaf. If you liked this video, then do not forget to like and comment. Subscribe to my channel to watch upcoming videos.